Let's talk about cells and tissues of the immune system. What is hematopoiesis? Hematopoiesis is the formation and development of red and white blood cells. What are stem cells? Stem cells are cells that can differentiate into other cell types. The cellular mediators of innate and adaptive immune systems are derived from a common hematopoietic stem cell. In the bone marrow, we have a pluripotent hematopoietic stem cell, which then results in the bone marrow producing a common lymphoid progenitor and common myeloid progenitor. What is the result of common lymphoid progenitors? In the blood, this would result in B cells and T cells. B cells would result in what type of effector cells? B cells would result in plasma cells. T cells would result in what type of effector cells? T cells would result in activated T cells. Within the bone marrow, a common myeloid progenitor would become a granulocyte or macrophage progenitor. In the blood, this would result in granulocytes. What are examples of granulocytes? Examples of granulocytes would be neutrophils, eosinophils, and basophils. Within the bone marrow, a megakaryocyte or erythrocyte progenitor would result in a megakaryocyte, which would then result in platelets. An erythroblast would then result in an erythrocyte. In the tissues, what type of cells would result from monocytes? Examples of cells resulting from monocytes would be mast cells, macrophages, and immature dendritic cells. What are Langerhans cells? Langerhans cells are dendritic cells or antigen-presenting immune cells of the skin and mucosa and contain large granules called Berbeck granules. They are present in all layers of the epidermis, but are most prominent in the stratum spinosum. Let's talk now about the cellular components of immunity. Basically, what are the cells that are involved in adaptive immune responses? These cells are antigen-specific lymphocytes, also specialized accessory cells that participate in lymphocyte activation, and effector cells that function to eliminate antigens. Describe the principal functions of the lymphocytes, B lymphocytes, T lymphocytes, and natural killer cells. Remember that natural killer cells are functioning cells of the innate immune system. B lymphocytes are mediators of humoral immunity. T lymphocytes are mediators of cell-mediated immunity. And remember that natural killer cells are cells of innate immunity. Describe the principal function of antigen-presenting cells, dendritic cells, macrophages, and follicular dendritic cells. Remember that the antigen-presenting cells capture antigens for display for lymphocytes. First, dendritic cells. Dendritic cells are concerned with the initiation of T cell responses. Macrophages are concerned with the initiation and effector phase of cell-mediated immunity. Follicular dendritic cells are concerned with the display of antigens for B lymphocytes in humoral immune responses. So as a review here, which are concerned with the initiation and effector phase of cell-mediated immunity. Macrophages, which are concerned with the initiation of T cell responses. Dendritic cells, which are concerned with the display of antigens for B lymphocytes in humoral immune responses. Follicular dendritic cells. Describe the principal function of the effector cells, T lymphocytes, macrophages, and granulocytes. Remember that the effector cells are for the elimination of antigens. When you talk about effector cells, we're talking about the way that they kill. First, T lymphocytes. These are concerned with helper T cells and cytotoxic T lymphocytes. Next, macrophages and monocytes. These are cells of the mononuclear phagocyte system, and granulocytes, such as neutrophils, eosinophils, and basophils. The mnemonic to remember granulocytes is BEN, basophils, eosinophils, and neutrophils. So once again, let's review which kind of cells present to which kind of cells. Follicular dendritic cells present antigen to B cells, dendritic cells present to T cells, and macrophages are concerned with the initiation and effector stage of cell-mediated immunity. And also know that B cells can present to T cells. Now let's talk a little bit more detail about lymphocytes. Where do lymphocytes circulate? Lymphocytes circulate continuously in the blood and lymph and are capable of migrating into the tissue spaces and lymphoid organs. What are the three main cells concerned with immunity that are called lymphocytes? These are T lymphocytes and B lymphocytes and natural killer cells. Remember that natural killer cells are part of the innate immunity. Lymphocytes look very similar, so how can we tell the difference? We can differentiate the cells based on surface proteins called CDs, or cluster of differentiation markers. For example, CD4 T cells and CD8 T cells and CD19 B cells. We know that B lymphocytes mature in the bone marrow and that T cytes would mature in the thymus. Remember also that B lymphocytes have a membrane-bound antibody as their antigen receptors. Each has an identical binding site for antigen. Activation causes naive B cells to differentiate into effector cells called plasma cells and into memory B cells. Remember that plasma cells secrete antibody. Now let's talk about T lymphocytes. Remember that T lymphocytes mature in the thymus, and they have T cell receptors, or TCRs, and these only recognize antigens that are bound to major histocompatibility complex, or MHC. So we have to think about antigen presentation. What are the subpopulation of T cells that help the activation of B cells? These are CD4 cells. What are the subpopulation of T cells that can kill virally infected cells? These are C cytotoxic cells, or CD8 cells. So remember that an immature T cell can then mature into a mature helper T cell, or CD4 cell, or to a mature cytotoxic T cell, or CD8 cell. Later we will talk in detail about the T cell receptors and the dendritic cells with the MHC complex, but just now to give a basic summary, the T cell would have the CD28, which would meet with the dendritic cell B7, and also the dendritic cell would have the CD40, which would meet with the T cell's CD40 ligand. Those would be secondary stimulators, but the first stimulator would be, or the first signal would be the T cell receptor on the T cell and the MHC major histocompatibility complex on the dendritic cell. We'll talk about that more in detail later.